To make a long story short, I've been working, I'm an artist working in emerging forms, and I've been working with DNA now basically for about the last 12 years. And the way I work with DNA is probably different than you might expect. I'm not interested in DNA, like just representing DNA or genetics via some kind of pictorial form. I'm not interested in just sort of going into a laboratory and saying, you know, take my DNA, make me a genetic portrait, and then recontextualizing it in the space of an art gallery. I'm interested in it as a really dynamic material form, and something that has no inherent, an image that has no inherent form. So for instance, an image that has been sort of reified as kind of being an essence, but is not really an essence. I thought before going deeply into my own work, this field has come to be known as bioart. And I thought I would, oh no, everything's going the wrong way. I thought I would start looking at the field by examples that might like be particularly sort of hackle-raising, and the works are probably, I think, the most misunderstood in some ways. Theorist Simon Penny has said that the task of contemporary art is to produce a highly charged ambivalence in the viewer. You know, to leave them stuck between how to read, you know, the work itself. But this work is called Victimless Leather, and it's by a team, a duo named Oran Katz and Yonat Zur, that go under the kind of uncatchy title of the Tissue Culture and Art Project, they're known as. Well, what they do, I think, better than anyone is I think, put one in these ethical conundrums. So, for instance, in this piece called Victimless Leather, we have what they call a piece of semi-living art. Semi-living because it is a cell mass sitting in an in vitro solution, a bioreactor. What's clever, I think, about this work is, on the one hand, we look at it and we say, oh yes, this is victimless. This is a piece of meat that was created from a dead cow, basically taken from the bone marrow cells of a piece of meat in a butcher shelf and used to grow synthetic, you know, grow in vitro leather. So supposedly victimless, although as one comes to learn in any of their workshops or any of their performances or any of their talks, there are all kinds of hidden victims in vitro processes. For instance, the fetal bovine serum that is used to keep these things alive is involves all kinds of fetal pigs kept in sterile conditions, which nobody really wants to know about. Also, I think really important in their work is another term that they coined, which is called the aesthetics of disappointment. So this little jacket is actually only about half an inch, about the size of a dime. And one of the things that they try to do, again, is looking at these kind of hyperboles of the, this is not even genomics, but the sort of hyperboles of the biotechnological institutions. And they try to really humble our expectations. So this little half-inch jacket. Another artist, again, who I think is easy to take the wrong way is, and people probably know this work. Do people know this stuff? Raise your ear arm or something. Again, I think this is highly spectacular work, spectacular in all connotations, by the artist Stellark, also an Australian artist. And I think simply read, Stellark's work can, again, seem really irresponsible. In this case, Stellark has inserted basically a cellular scaffold beneath his skin, very similar to the scaffold that Katz and Zur use to build their jackets or their sort of flesh sculptures. He's inserted it beneath his arm. And if you take him at face value, Stellark says, well, this was, the human body is obsolete. And we need to be extending our senses in all this way if we are to progress as a species, which sounds very transhumanist and naive and techno-utopian and all of this. But hardly anybody ever does. Well, first of all, I've once asked my students, how would you extend your senses? And nobody's ever imagined extending the senses through hearing. It's always some kind of an ocular escalation that involves, seeing broader spectrums, or maybe seeing further, or having eyes in different places. Never the ears. So where does the ear come from? right? I don't know if people remember in the mid to late 90s, this is the famed Vacanti mouse. There was kind of I guess, a sort of press PR move of Vacanti's Harvard lab to show the kind of efficacies of the things that could be done with tissue culture. Of course, the mouse doesn't live very long. The ear is never operational. It is just like Katzenzer and just like Stellark's work. It is a scaffold stuffed under the skin, which its own cells take over, which it doesn't reject because it's a specialized oncomouse, which has no immune system. Anyways, I bring these things up because oh, and I guess lastly I should say that when Stellark describes his work, he, for instance, his ear on arm, he seldom describes just how useful it is. He mostly likes to talk about his first trip to the doctor's office when it was contaminated, and he shows up and says, "Doctor, I think I have an ear infection." Again, so this work, I think, looks, these previous examples of bioart, they look kind of sensational. I think actually under the surface of them all, what's interesting is that they're messy, right? That, unlike the kind of clean presentation we see in a lot of biotech, they're messy, they're dirty, they don't quite work very well, and there's a kind of deep critique embedded. And I think the thing that's going to be really maybe a problem for bioart is the emergence of this synthetic biology as a kind of tantalizing carrot that's been, I think, dangled in front of artists. I think some of the things that are kind of frightening about it are the ways in which, as it's promoted for artists, it tends to always be a playful tool. And I think this is indeed technology that they're looking for artists to help normalize. I first got into my work really via the sort of ELSI symposium, or not a symposium, a roundtable at the University of Washington, Seattle. It was organized to introduce artists to geneticists and genomicists. That's where I met Mary Claire King and Maynard Olson, and my first project wound up being a collaboration with the King Lab, actually. You're familiar with this project, so I won't describe it. I'll tell you the thing that interested me. Shortly after this roundtable, the rough draft of the Human Genome Project was completed, and the scientists said, there's no scientific basis for race. The issue is, of course, a concern because the original title for the Human Genome Project was Neo-Eugenics, which James Watson and probably others suggested that the name might carry negative connotations. So they suggested it be changed. However, I felt that probably race, we could say there's no such thing as race, but racism would not disappear. That in fact racism would go molecular. That the very deviants that were formerly kind of lodged in our skin, or skin as signifier, would now be more deeply encoded in our DNA. That it wouldn't disappear. That we would have a new molecular racism. And as a kind of as a way of understanding how that might work, I looked back at Charles Davenport's early 1929 study called Race Crossing in Jamaica, which was a high-profile eugenics study, basically showing the inferiority of hybrid browns compared to their white and black parents. I have this interesting, I'm also a descendant of hybrid Jamaican families. My mother coming here from Jamaica in 1962 or something thereabouts. So I sort of proposed a very perverse project. The idea was to kind of look at some of these metaphors of the new genetics and the old genetics, particularly the term genetic fitness, and to sort of recreate how genetic fitness might be measured in the genomic era. It would no longer have to use anthropomorphic measurements to be measured. It would use something far more seductive. So what I did was I worked with the King Lab and I extracted skin color genes from my family members. That was previously my sister there. And the idea here was, again, to rethink this medium of gel electrophoresis. How could this work if, in fact, it was a plastic media that could be used in all kinds of ways to inform but also to misinform? So in this first case, what I wound up doing was building this kind of fully live experiment that would run in a gallery 24 hours a day. successively over the course of however long, all mechanized. And the idea was that the DNA would go into the lanes of the gel, these 4 lanes, skin color genes extracted from each family member, mother, father, sister, brother, and I would race them against one another. So the idea was a race about race in which the bodies had actually been erased. So these contestants moving against each other, and it would all be fully automated, and a computer would be doing machine vision algorithms to determine where DNA was. So this very kind of high-tech spectacle, which of course everybody knows has no meaning, because the way that electrophoresis works is that DNA of different sizes migrate at different rates. The smaller fragments move the fastest. So in fact, the winners of this race were just those that had the smallest skin color genes, nothing more than that. Unless you'd like to read more into it. This was the kind of conundrum that this work put forth. It was created in 2002, and I called it the relative velocity inscription device, because it looked at the velocity of relatives to determine their fitness. There's a close-up of the screen. The figures, finally somebody really found this out and wrote about. The figures are actually Muybridge figures, because Muybridge is also kind of part of that whole eugenic notion of understanding bodies through anthropomorphic measurements. Now, what was curious is that piece I finished in February of 2002. In 2001, the Sparky Nobel laureate James Watson, in his famous lecture at UC Berkeley, said this interesting thing. When you make a piece like this as an artist and you're coming into a new field, you don't necessarily know if your sort of very critical worldview is really what's going to happen. I mean, I was thinking, well, maybe I should be more optimistic. Maybe race really did go away. Well, Watson, of course, at that experiment describes this experiment of his colleague at the University of Arizona, intended to test whether he could chemically darken the men's skin as a skin cancer protection, only to observe this unusual side effect. The men develop these sustained and, of course, unprovoked directions from the melanin treatment. And then he goes on to say this melanin injection is even better than Viagra, because you don't even have to think about sex. That's why you have Latin lovers and only English patients. Which is, of course, very funny. And yet, at the same time, it truly shows that the racism has gone molecular, right? That now we can demonize these very genes in our bodies that produce this melanin substance, and that's actually the root of all these stereotypes, which we had thrown out and called cultural until Watson attempts to bring them back. Anyways, from this, I got really fascinated with these different kind of metaphors permeating this medium, and particularly this one, the DNA fingerprint. And I thought, wow, that's interesting, because it's not a print. It's got nothing to do with a print. It's not an imprint. It's produced in a laboratory. And why that name? And then I started to also think that this is the most authoritative cultural image there is. This can put a person at the scene of any crime. It can put a person behind bars. This really deserves some investigation. So I looked, I looked at who was talking about this term. One of the people being the head man at the FBI labs, who said, we like the term DNA fingerprinting because it cements in the mind of a jury that we're identifying one individual to the exclusion of all others. Well, that's nice. It cements in the mind of a jury. That's a good reason. The inventor of the technology, Alec Jeffreys, says later, if we could call it anything else, nobody would have cared. Call it DNA fingerprinting and the penny dropped. Which brings to mind this illustration from Bruno Latour in a Science and Action text, the old face of science saying, once the machine works, people will be convinced. When in fact, the younger face says, the machine will work when all the relevant people are convinced. In the case of DNA, it was basically the juries. In the case of the term DNA fingerprint, they were the people that really had to buy into it, how well it worked. And the metaphor really helped that out. I went on from this to make another piece. This piece is called Latent Figure Protocol. And the assumption, the piece worked out, this was the rationale. There's no such thing as a kind of singular DNA fingerprint. In fact, there are hundreds of thousands of different ways to make a fingerprint. Because part of the process is the use of chemical enzymes. There are hundreds of them. They can be used in all kinds of different combinations. Each one attaches to a different recognition sequence in your DNA, cuts it in different places. And these kind of things, the interplay of these things are responsible for the banding pattern that a DNA fingerprint takes. Then I thought, well, okay, what should be possible then is to do molecular biology in reverse, right? A kind of critical molecular biology, like almost like a counter laboratory of molecular biology, in which, starting with an organism, I could, for instance, target regions of that organism. This is called a plasmid. And simply by applying the right enzymes to the DNA of that organism, I could cut that DNA in certain spots which I would know in advance. In this kind of case, I should be able to make almost whatever image I wanted. DNA works like this, that you have these fragments, you insert it in picture B into a well of a DNA gel, you apply voltage, and the small ones move faster than the big ones. And they move at a rate inversely proportional to their size, and a rate which could be empirically discovered. It wasn't difficult. Then from there I went on and I wrote a computer program through which I could say, okay, let me put an image into this program, and then let me try to basically come up with a recipe through which, for any given organism, I could make their DNA look like whatever I felt like. So this was the latent figure protocol simulator. And from that, then, I got this idea that what needed to happen, unlike the crime labs, the DNA fingerprint happens behind closed doors, right? And we're not told anything about the enzymes used, any of this kind of thing. I thought the whole process needs to happen live. You need to do it in front of audiences. It needs to be performed. And they need to see that image emerge. So I always do 2 images at a time, because the idea here is to show not only that the DNA image can be manipulated to form an image, but moreover, to explain that very much like we have no one singular DNA image identity. The DNA image is not essential to us. It's not part of us. It's not sitting latent within our cells. It's something that's made in a laboratory. And there are all these different images that I made in the series. I always include the recipes on them, so that just like a proper scientific proof, if somebody wants to check my work, they can. All the information's there if they want to try to do it themselves, or to disprove me. Skull and crossbones, for those of you. I'll go back, so that's the skull and crossbones. Anyways, so the DNA image then it's been my fascination. I'm going to show one more piece, which is called the Suspect Inversion Center. And I'll get to that now. I was always fascinated by this famous chase that you might recognize from 1995. It's always been important to me. And let me grab my notes. Interestingly enough, Eric Lander stated that the US public, usually indifferent to matters scientific, has become obsessed with DNA. Even tabloids offer commentary on restriction fragment length polymorphisms. And this is not stemming from new genetic breakthroughs, but the O.J. Simpson case. But what was interesting was that I thought, okay, if this is all happening, I should really find this original DNA document and just come to an understanding of the case via the document. So I searched all over for where this might be. Finally, I came across the O.J. Simpson Forensics Archives at Cornell University. And I took a visit. I found lots of the image. Here it is, if the genes fit. I found this in dozens of papers in the O.J. Simpson Archives. Of course, it's an image from Cellmark. Cellmark was the company that did the work for the prosecution. But we don't see here the actual O.J. Simpson image. We see basically a promotional image that shows how easy it is to read a DNA image. So I called up some of the people from the O.J. Simpson defense team. A guy named Bill Thompson was the first at UC Irvine, and I said, well, why is it that we never saw the actual image? Was it a matter of privacy? What's behind it? And he said, we kept trying to give the press the image, but they just didn't care. They wanted this to be a simple thing about juries being confused or, you know, post-Rodney King bias or something like this. But in fact, we tried to get them to see it, but they weren't interested in the bigger, more complicated story. To make a long story short, I tracked down this image at the Smithsonian National Archives, I guess where Charlie also got that work, got his work kind of immortalized. I finally found the image, and I thought it was intriguing. I didn't quite know what to do with it. But I thought what really needed to happen, there was so much obfuscation of things in the O.J. Simpson trial. On the one hand, we had these sexy vocabulary, but we weren't really learning anything about DNA. Supposedly the DNA wars have been won, and you know, Barry Scheck was part of the O.J. Simpson defense team. So there's some really interesting things happening in that place. But we didn't learn much about them, and we certainly didn't really learn about how DNA evidence is made. So I set up a project called the Suspect Inversion Center. Suspect Inversion Center is it's an acronym for CSI, or an anagram of CSI, but it's, the idea was it should also be a functional inversion of CSI. So the Suspect Inversion Center was an operational laboratory engaged in reproducing historically important DNA artifacts. In this first instance, reproducing an entire O.J. Simpson trial DNA image, showing victims as well as suspects' DNA, rendered using my own DNA. So the idea was to use my own DNA to recreate this historical document. And the premise was that the history of art is replete with this notion of the copy. For instance, there's the master study, there's art forgeries, and there's pop and postmodern appropriation. Each which has different kind of effects on the original and different kind of levels of reference and destruction of certain auras. So we went to work over the course of 6 weeks recreating it. We still haven't done it perfectly. This is my assistant, Carrie Sullivan, who's now going to law school, but she was my assistant in the project for 2 years. Basically what we did was we kind of reverse engineered the original image, looking at the different sizes of every different DNA band, and then having to recreate mine through a PCR process. And these are some of the different steps along the way. These are some of the different process shots, and we got quite close. And the whole idea was that we'd also be performing this live, demystified, you know, in public space. This was a gallery in Berlin that hosted the project for 6 weeks. And so the idea was that this would be kind of as open a process as we could possibly have as we took on this sort of very kind of reverential task of reproducing the image. At the same time, we wanted to also have this discourse with people present that, that eradicated the difference between merely technical details and basically cultural details of the case. This doesn't mean this isn't my own work. I just wanted to say that it's been great being here at the conference, and I put this up because I think this is an occasion to say, you know, if there's any way we can collaborate, I'd, you know, be happy to. All kinds of opportunities in this work.